Hello students, today I'll be introducing ProtGPT2, an AI model inspired by natural language processing specifically designed for understanding and generating protein sequences. We'll explore its applications, key concepts, and then we'll dive into an exciting demo where we'll work with real protein sequences and use this ProtGPT2 model to generate novel protein sequences, and then we'll also visualize them using AlphaFold server provided by Google. Proteins are at the heart of life itself. They play a critical role in everything from metabolism to DNA repair. And this question to why study proteins with AI is very easy to answer today, because traditional methods for studying and designing proteins are very labor intensive and often requires expert knowledge and intensive experimental setup. So by applying AI, specifically these large language models like ProtGPT2, we can drastically accelerate how we analyze and design proteins, which opens up new frontiers in biology and medicine. A recent example of how important role AI is playing in understanding proteins is AlphaFold sharing the Nobel Prize in Chemistry. So this ProtGPT2 is a deep unsupervised language model for protein design. It's built on the same foundation as GPT models for human text. But instead of learning the human language, it learns the language of proteins. It is trained on protein sequences. ProtGPT2 understands the grammar and syntax of the proteins. It captures the underlying biological principles like structure and functions. Okay, this model treats protein sequences as sentences in a language. Just as a GPT model predicts the next word in a sequence, it predicts the next amino acid in a sequence. Now, ProtGPT2 predicts the next amino acid in a sequence of proteins. This approach helps it learn the relationship between the residues, the secondary structure, and even the functional motifs, just to caught the relationship between our natural languages and this protein design, the similarity between these. I'll quote a sentence from the paper itself. So this is the peer-reviewed paper of ProtGPT2. So here if you see, they mention that protein sequences can be described as a concatenation of letters from chemically defined alphabet, which is natural amino acids. And like human languages, these letters arrange to form secondary structure, which in our case is words, which assemble to form domains, which in our language are called sentences, that undertake a function, which in our language is meaning. And also one of the most attractive similarity is that protein sequences, like our natural languages, are information complete, which means they store the structure and function entirely in their amino acid order with extreme efficiency. So these analogy between protein sequences and human languages have also been noted by others and authors of this paper, which motivated them to study protein sequences using large language models. Okay. Next, I'm going to give you an overview of the demo. So first, what we'll do is we'll get a protein sequence from a protein database like UniProt. Then we'll use this ProtGPT2 model to generate and extend the given protein sequence to generate a new protein sequence. Then we'll visualize the generated protein sequence as well as the original protein sequence using the AlphaFold server provided by Google. We'll also see, since the inference time of this model is going to be very fast, we'll see how we can generate multiple sequences by giving a single input, and then how to select the best sequences out of all. So that is about the demo. Okay, but before starting the demo, I would like to acknowledge the developers of ProtGPT2, AlphaFold server by Google, and Hugging Face and Biopython libraries for making this demo possible. Okay, now let's go ahead and jump straight to the demo. Okay, well, let me search for ProtGPT2 Hugging Face. this model is present on Hugging Face. Okay. Okay, in the first line itself, they also share the peer-reviewed paper, which I would encourage you to go ahead and read. Okay, coming back to Hugging Face. So there is a description of how to install and use. This is a detailed description. But today we are going to use Google Colab for our purpose. So I'm searching Google Colab. Okay, I'll create a new notebook. So this might take a while. Okay, so we have this new notebook. Let's name it ProtGPT2. Okay, that name is just ProtGPT2. Okay, so the first thing we'll need is the libraries that are going to be useful, and let's install them. The first one is transformers library, since we are working with a deep learning model. Let's pip install transformers. Okay, and the next thing is Biopython library. Biopython. Okay. Okay, since while these dependencies are being installed, let's look at other libraries that we will need for this demo. The first thing obviously is going to be a tokenizer, since, okay, why would we need tokenizer that we can import from transformers? Since the input is a protein sequence, but the model takes input in form of tokens, not in the text or protein sequence. So we'll need a tokenizer to convert our input sequence into the form of tokens. So let's import from transformers. Let's import Auto-tokenizer, and we'll also import auto model for causal LM. So this auto model for causal LM will use to load our pre-trained model, which is obviously an autoregressive model or causal language model. Okay, sorry. So since we have imported these dependencies, let me create a tokenizer. This tokenizer will use auto-tokenizer dot from pre-trained, since we are using the pre-trained model as well. Later in the demo, I'll also show you how we can fine-tune the pre-trained model on selected sequences. Okay, but before that, let's write the model name as well here. Model name. Sorry. The model name is, we can get the model name from here. You can always copy model name from Hugging Face like this. So, okay, this is a model name. Okay, next thing is let's import our model from auto model for causal LM, again dot from pre-trained and model name. Okay, so by far what we have done is we have created a tokenizer instance which have the information specific to this particular model name. Then we have created a model instance by importing this ProtGPT2 model using auto model for causal LM. Okay, the next thing that we would need is a device to, okay, next thing we are going to put this model into a device, and for that we'll need Torch, that is import Torch. Okay, so Torch might be familiar to most of you working in machine learning or deep learning. It is an important library, a core library of Pytorch, which is an open source machine learning framework by Facebook. So let's define our device, which is going to be, torch.device. And since we are using a large language model and we would need GPU, so the model will be CUDA if, but it is only if we have GPU available. So we'll write torch.CUDA.is available. So this is a function which will return true if GPU is available and false otherwise. In else case, we'll use CPU as the device. And next thing is to model.toDevice. So this part of the code is moving the model dependencies and model computations to the device. The device would be CUDA or CPU. Okay, it shows some error. Okay, error is gone. Okay, but there's one thing that I forgot to mention here, which is by default the device on a Google Colab notebook is going to be CPU, which you can change by going to runtime and then click on change runtime. Here instead of CPU you can select GPU. Okay, so I've selected GPU, click on save. Okay, so let's run. Okay, so meanwhile this is running, let's write our input sequence. So we'll get our input sequence from a protein database like UniProt. So let me search for UniProt. Okay. So I'll get, let's say, a protein HBB, which is hemoglobin subunit beta. So let me get it for human. I can click on download. And I can download it in FASTA format, but since this is a small protein sequence of 147 amino acids only, I'll click on preview and just copy it from here. Okay. So our code to load the model tokenizer and moving the model to device is running. Meanwhile, let me just indent the sequence well. Okay, but since a model would require the input in form of tokens instead of a sequence of text or amino acid, we'll have to get that into a particular format, which is going to be input, and we'll utilize tokenizer for that again. So tokenizer, and this tokenizer would expect the input sequence. And it would also expect a variable or a parameter to specify what is the type of tensor it is requiring, whether it is, let's say, TensorFlow or Pytorch. So we'll define that by return tensors to be PT, because in this case it's going to be Pytorch. And again the last part, again will move the input tokens again to the device. So okay, let's run this code to ensure that our input is created in the form of tokens. You can also print it, and this no longer is going to be in form of this text but in form of these input, where the first is the input IDs, which is also going to be a parameter for model.generate function, which we are going to generate new sequences. So let's go ahead and do that. We'll define a variable output, which is model.generate, and then as I mentioned, we'll define input IDs. Other things that we need to define is the max length. Though this is an optional parameter, you can define the maximum length you want to extend the given protein sequence. So let's write 100 here. And then again, it's a number of sequences you want to get as output. As I mentioned, since the inference time of this model is very fast, you can define the number of sequences you want to generate given a particular input sequence. So let me write num return sequences, which is going to be, let's say, 1 in our case. We'll also see an example that we can define 100 or more than 100 here. Again, do sample, yes, true. And then top k, which is just a strategy. Let me write, let's say, some 950 here. So this top k is a, there is some error shown by this. Just write num return sequences. Oh, it's right, it might go. Okay. So top k is just a strategy of randomly sampling. So instead of randomly sampling from all available tokens, this says that randomly sample from top 950 most probable tokens, right? So you can tweak this parameter as well. Other parameters which I'll show you here, you can also define like a repetition penalty or number of returning sequences, or again other parameters. Okay, now let's go ahead. So this will generate the output, but again, as I mentioned, this output is not going to be in form of protein sequence, but it is going to be in the form of tokens. So let's make another variable named generated sequences, and again we'll use tokenizer to decode, and this will take input output 0, and we can just skip the special characters, skip special tokens. We'll set this to true. Okay, so we have this generated sequence. So now let's go ahead and print a generated sequence. Generated sequence. Okay. So it shows some error. Okay, there is a spelling error of generated sequence. I'll just copy paste. As I mentioned, the inference time is going to be very fast, so it has generated sequence in a matter of seconds. So, okay, the next thing that we are going to do is go ahead and use AlphaFold server to visualize these protein sequences. So first of all, we are going to visualize our original HBB protein sequence. Okay, so let me copy the sequence. Okay, you can also define the entry type to be DNA, RNA, or protein. In our case, it's protein, so we'll define protein. Next thing is number of copies. If you increase number of copies to be 2, 3, or 4, you will also see the interactions between these two copies, how they kind of coexist. For a demo purpose, I'll keep the number of copies to 1. You can also define the job name, which this is original HBB, and I'll confirm and submit the job. You'll see the jobs running here. Meanwhile, this job is running, let's submit another job, which is generating the 3D structure for the generated sequence. Okay, for better visualization purpose, let me open it into a different tab so that we can co-visualize both of them. Okay, define it, let me indent it well, and let me write it name generated sequence. Okay, let me submit the job. So both of these jobs are now running. This might take a while. So let's do another thing, which is let's try to generate a random protein sequence of size, let's say 150 or 200, and see how does that perform when we try to visualize its 3D structure using AlphaFold, because that will give us an idea that the generated sequence by the model are not just some random sequences, but they do have some meaning and they fold into a stable structure as well. So let's import random. We'll need all these amino acid characters. So let me write amino acids. I already tried this demo once, so I have this helper function. So I'll get the amino acids from here. Okay. The next thing is we are going to generate a random sequence. So let's just say random sequence needs to be dot join. You can use multiple ways to get the sequence. Let's say random dot choice and select from this string amino acids, and I can also define the length that I need, which is going to be, let's select 150 in this case, or let's just say let's select 200. So we have a random sequence. So now let's try to print the random sequence. Okay, let's go ahead and run this code. Okay, there is some error. Okay, so this k parameter has to be inside this. So okay. Okay, this has to be choices, not choice. Okay, so we have a random sequence here. So let's go ahead and copy this random sequence. Let's create a new tab for full server. As you can see, both of these jobs are complete. But before checking the output, let's submit this job as well. And let's write, let's say random sequence, right? Confirm and submit the job. Meanwhile, let's have a look at the output of original HPB. Okay, so you can see, so there are two things that you can look at. One is this PLDDT, which is atom-wise confidence score. Higher the PLDDT value, more confidence the part of the structure has. For example, the darker blue is high confidence, this yellow part low confidence, and then this PTM value gives you an idea of the overall structure confidence, and generally a PTM value of greater than 0.5 gives you the idea that the structure of this particular sequence is close to the original sequence maybe. So it's the generated structure is close to the true structure. Let's have a look at the generated as well. Okay, so it's not, the PTM value is not 0.9, but it's again pretty much higher than 0.5. So this is the structure for our generated sequence. Now let's see if the random sequence is complete. We'll have to wait for a while so that the execution of this random sequence 3D structure generation is also complete. Meanwhile, since this is taking longer than usual, let me go ahead and look at some of the other things that are present in the Hugging Face repository of this ProtGPT2 model. So we did this de novo proteins, generation of de novo proteins in zero-shot fashion. We didn't do any fine-tuning on selected sequences, but you can do fine-tuning on user-defined sequences, and a template code is present on this Hugging Face repository. And also one thing that I mentioned is, since the inference time is very fast, you can generate a lot of sequences, maybe 100 or 1000 sequences in one go, and then select the best. So there is a section of how to select the best sequence as well. So to select the best sequence, you can calculate perplexity for each of the sequence generated. So this perplexity function just takes the sequence, the model, and tokenizer. So for example, if I show you from here, so you can just do a for loop on this. For example, let me just go ahead and demonstrate that. For example, let's say for sick and out this, for example, for sequence in, let's say, generated sequences, but instead of defining 0 here, I'll just do this, and for each of the sequence, for each of these sequence, I'll pass it to the perplexity function, and we'll calculate the perplexity value. The authors does not specify a particular threshold to call a sequence to be good or bad, but you can always go ahead and generate multiple sequences and select the best one. The lower the perplexity, the better the sequence. The training specs of this particular model are also mentioned on the Hugging Face repository. Okay, the random sequence is also generated. Let's have a look at that. Okay, so here if you see the PLDDT score is very low. It's you don't see the dark blue or yellow color. It's quite orange. So which means that the model does something. It's not. You cannot generate a random protein sequence and get a good structure or a meaningful sequence by generating random sequences. So this is just a visualization for better understanding and to see how to evaluate the accuracy of a model. I would encourage you to go ahead and read this paper. This paper is open access. You can read. And even if you do not have a good background in machine learning and deep learning, I encourage you to read at least the introduction, the results and discussion section of this paper to get a high level idea of how things are working. So going back to our presentation. So the key takeaways from this demo was to demonstrate how to use open source large language model. For example, this ProtGPT2 has many applications starting from drug discovery and synthetic biology, in disease research as well, because you can generate proteins that maybe bind even better to a specific site than the original proteins that we have explored. If you go and read the paper, you'll get more insights about this. But as like any other large language model, there are some limitations because a large language model depends on the training quality, like the training data quality, and it may also require fine-tuning for specific purposes. Thank you, and I hope you got a hands-on experience on using a large language model. I encourage you to try this yourself or some other model like DNA BERT. Thank you again.